This is the second part of my video on genetics, and in this part I would be covering up basic genetics and the various terminologies associated with it. So let's begin. There are millions of species living on Earth, and they possess millions of different traits and characteristics, most of them inherited from their parents, and some of them are new. So let's zero down to Homo sapiens. Now, the human body consists of millions of cells, and each of these cells has a nucleus. And deep inside the nucleus is an amazing molecule which is called as DNA, or the deoxyribonucleic acid. Sections of this DNA are called as genes, and together all genes in the body make up the genome. So what exactly is the genome? The total genetic makeup of an individual is known as the genome. It is functionally divided into genes, and in human it is about 25,000 to 30,000 different genes that make up the genome. The genome is split into 23 pairs of chromosomes, and each chromosome contains a long strand of DNA that is tightly packaged around certain proteins which are known as histones. So what exactly are chromosomes? They are thread-like structures located inside the nucleus, made up of proteins, that is nothing but histones, and a double helix of deoxyribonucleic acid. Now these chromosomes have two regions or areas. One is known as the heterochromatin. These areas of chromosomes look thick, coiled and condensed, whereas certain other areas of the chromosome resemble thin or lightly stained threads which are called as the euchromatin. They potentially contain the active gene component, and the identified genome sequence represents around 90% of euchromatin. Let's go on to learn the structure of each of these chromosomes. Each metaphase chromosome comprises of two identical components, and these two symmetrical halves are called as sister chromatids. Now we can see two sister chromatids here. Let us consider one out of this. In the center, they are attached together at a constricted region which is called as a centromere, as you can see here, the centromere. Now the centromere divides the chromosome into a short arm which is represented by the letter P. This is the short arm, and a long arm which is represented by the letter Q. Now the ends of these chromosomes are known as telomeres, the ends of both the sister chromatids. Now there are certain areas in the chromosome which display a banded pattern when they are treated with certain stains. That is nothing but alternative dark and light bands that appear along the length of a chromosome, as you can see here. Now these are useful to identify certain chromosomal aberrations such as breakages, loss, displacement, translocation, inversion, etc. Now let's see how this cytogenic map location helps us in identifying these aberrations. So basically a chromosome is numbered. Every chromosome, all the 23 chromosomes, every chromosome has a number starting from 1 to 23. Each of the chromosomal arm, like one sister chromatid, again is split into a short arm and a long arm. Short arm represented by P, long arm represented by Q. Each of these arms again are divided into certain regions, like you can see here, P arm into 1, 2 and 3, Q into 1, 2, 3, 4 and so on. Each of this region is again further divided into bands, like band 1, band 2 of this region, and each of this band is further subdivided into sub-bands like this. So supposing we take up a cytogenic mapping location as 3p22.1, 3 is the chromosomal number, P is whether the short arm or the long arm, as in this case it is the short arm. 2, the first 2 represents the region of the chromosome, the second 2 is the band of that region, and the final number represents the sub-band of the band. So I hope I've made myself clear. Now this system enables us to accurately identify even small structural anomalies within the smallest subdivisions of the segments of chromosome at its exact location. Going on to what is DNA. DNA is deoxyribonucleic acid. It is the blueprint of life, consisting of a double helix of two polynucleotide chains that run anti-parallel, and this model was first established by James Watson and Francis Crick. Now let us try knowing about the basic building block of a nucleic acid, that is the deoxyribonucleic acid in this case. This is nothing but a nucleotide which makes the building block, or which forms the nucleic acid. Now, the nucleotide has three components. It has a nitrogenous base, it has a deoxyribose sugar, and a phosphate group. Now, the nitrogenous bases can either be purines or pyrimidines, like they can either be adenine, guanine, which are purines, or cytosine, thymine, which are pyrimidines. Then apart from this, the polynucleotide chain is constructed by linking the 5' end of one pentose ring to the 3' end of the other pentose ring by phosphodiester bonds. Also, the adenine, the bases pair with each other. Adenine always pairs with thymine, and cytosine always pairs with guanine. Adenine and thymine share a double bond of hydrogen, whereas guanine and cytosine form a triple hydrogen bond. Now, if you consider one complete turn of the helix, that is 360 degrees, starting from here up to here, it contains around 10 base pairs of nucleotides, and the distance is around 3.4 millimeters in length. It consists of a minor groove, as you can see here, and it also consists of a major groove. This is what a double helical structure looks like and is made up of. Now, a single segment of DNA, sections of this DNA, are nothing but genes, and these genes, they code for one complete functional polypeptide chain or one complete functional protein which carries out the function in the body. So what is a gene? Gene is a sequence of DNA that codes for RNA. Each gene is a sequence within the nucleic acid that represents a single protein. Now, this word was derived from another word called pangene, which means to give birth or to become, and gene was derived from this word pangene by Johansen in 1909. Now, pangene was originally given by Darwin, but Johansen felt that using pangene as this pangene, that pangene would be an elaborate term, so he simplified that term by using the last three letters, and from then on it was called as gene. I have a small video to show how genes play a role in protein synthesis and how these proteins carry out the various functions in the body. So let's see with the help of a video. When a gene is switched on, an enzyme called the RNA polymerase attaches to the start of the gene. Now, it moves along the DNA, making a strand of mRNA out of the free bases in the nucleus. The DNA code determines the order in which the free bases are added to the mRNA, and this process is called transcription. Now, before the mRNA can be used as a template for the production of a protein, it needs to be processed. Now, this involves removing and adding of sections of RNA, and the mRNA then moves out of the nucleus into the cytoplasm. Protein factories in the cytoplasm called the ribosomes will go and bind onto the mRNA. The ribosomes read the code of mRNA to produce a chain made up of amino acids. Now, there are 20 different types of amino acids. The tRNA molecules, which are the transfer RNA molecules, they carry the amino acids to the ribosomes. Now, the mRNA is read 3 bases at a time. As each triplet is read, a tRNA delivers the corresponding amino acid. This is added to a growing chain of amino acids, and once the last amino acid has been added to the chain will fold and then form a complex three-dimensional shape to form the protein or the mature protein, which carries out the particular function of that gene. So basically what happens is the DNA undergoes transcription, wherein a particular segment of DNA is copied into the RNA, that is the messenger RNA. It is taking the message from the DNA by an enzyme, that is RNA polymerase. Then there is a process of translation, wherein the messenger RNA is decoded by the ribosome to produce a specific amino acid chain or a polypeptide chain. This polypeptide chain, once completed, would fold and form a three-dimensional structure into a mature protein. That protein carries out the particular function, and hence we see gene expression.